There are thousands of distinct enzymes in cells catalyzing billions of reactions. Each enzyme can speed up biochemical reactions mind-bogglingly fast. Extreme speed makes life possible, but strains our understanding to consider how each cell manages those billions of reactions every second. How does the cell control this enzyme activity? Unleashing such tremendous power without mechanisms to control it would create chaos. In this lecture, we'll examine the ways that cells regulate enzyme activity, exerting precise control over the synthesis and breakdown of biomolecules. Cells do this for every molecule they make, and they must do so continuously to maintain stable internal conditions called homeostasis. Now, one reason that biochemists care so much about the control of enzyme activity is that many medical conditions result from enzyme activity that's excessive or insufficient. By figuring out ways to inhibit or increase these reactions, we can treat the associated diseases. The Goldilocks principle applies to enzyme activity. You don't want too much, but you also don't want too little. It needs to be just right. If enzymes in your thyroid gland make too much hormone, you become irritable and sleepless. Too little, you're tired. These have serious consequences for your health. Enzymes are like automobiles with accelerator and brake pedals to adjust the speed appropriate to existing conditions. Now, not all of the thousands of reactions that enzymes catalyze are needed at any given moment. My pancreas synthesizes digestive enzymes to break down the food I eat. If those enzymes start working too soon, they act on my pancreas and I get pancreatitis, a potentially fatal condition. When I eat a meal, I need the same enzymes to break down the food I eat, or I get indigestion. Cells must sense levels of molecules and adjust actions on the fly. For example, suppose your muscle cells need glucose for energy to help you flee from a pursuing car salesperson. Something needs to turn on the enzyme that breaks down stored glycogen to release glucose. The glycogen breakdown enzyme must be active long enough to provide the necessary amount of glucose and then stop, or you would deplete all of your stored reserves. Now, reserves are important. You need them for emergencies. Other situations call for maintaining a steady supply of molecules used by cells on a regular basis. If levels of a particular nutrient are low within a cell, its synthesis must be triggered. But once levels of the molecule are back up, its production must be halted. This is kind of like the thermostat regulating the temperature in your house. A sensor triggers the heating if the house is too cold, and shuts it off when the correct temperature has been reached. Regulating production of biomolecules depends on controlling the activity of the enzymes involved. There must be something that senses the conditions, and then a way to switch the enzyme on or off. Cells use assembly lines to build molecules that they need. That is, there's an ordered sequence of reactions called a pathway, in which the product of reaction 1 is the substrate for reaction 2, and so on. In an assembly line, each step in making a widget is the responsibility of a particular worker who completes the task and then sends on the product to the next worker. Similarly, each reaction in a pathway is catalyzed by one enzyme, and the product of its activity is sent on to the next enzyme until the final product is obtained. The pathway to making steroid hormones, for example, has 30 such reactions, a long pathway. Chaining together multiple reactions into a single pathway gives cells a very simple way to regulate the pathway. Since each enzyme depends on substrate from the reaction before it, turning off one enzyme stops the activity of all those that come after it in the pathway. As a result, each enzyme in a pathway does not need to be micromanaged by the cell. Usually an enzyme early in the pathway is controlled by a mechanism called feedback inhibition. Now this makes sense. Let's say you wanted to stop a bakery assembly line that was making cakes because it had made as many cakes as you could sell that day. It's better to stop at step 1, where the flour is being measured out, rather than shutting off the process after the butter and eggs have been added. Stopping later in the process would leave you with batter you couldn't bake, and you'd be wasting ingredients that you could use to make something that you needed. However, the control is not always at the first enzyme in the pathway. Suppose an assembly line was making apple pies, and you had plenty of them. In that case, stopping after the crust was made might be more reasonable. Why? Because the same pie crust can be used for banana cream pies, which might be in low supply. Similarly, if a pathway can branch to make more than one final product, then stopping at the branch point makes more sense. Otherwise, shutting off the first enzyme in the pathway blocks the synthesis of all of the pathway products. Now, regardless of where the control point lies, metabolic pathways are always regulated, and that happens through the control of their enzymes. In the rest of this lecture, we'll examine some of the major ways by which the activity of enzymes is controlled. This includes the synthesis, allosteric, physical inhibition, and covalent modification. Now, I'll first mention that the crudest but most effective way to control enzymes is managing whether or not the enzyme is made by a cell at all. An example of an enzyme regulated in this way is chymotrypsin, which is made in the pancreas. Other types of cells in the body don't make this enzyme, saving themselves the work of having to regulate its activity. A simple yet much more nuanced mechanism for control, though, is called allosteric regulation. And it works like a dimmer on a light switch to adjust brightness. In this form of regulation, the pathway of a regulatory molecule in one part of the enzyme can adjust the enzyme's behavior. The binding of these regulatory molecules, called effectors, is reversible, allowing them to exert or remove control as needed. Now, we've encountered this phenomenon when we discussed hemoglobin and cooperativity. Though hemoglobin is not an enzyme, we saw that the binding of oxygen in one subunit of hemoglobin favored the R state, increasing oxygen affinity in the neighboring subunits. Conversely, binding of 2,3-BPG favored the T state, where hemoglobin's affinity for oxygen was reduced, allowing for the release of oxygen. So binding of a molecule in one part of hemoglobin influenced what happened elsewhere. So how does this apply to enzymes? Binding of specific regulatory molecules also affects enzymes as to whether they're in the R state, which favors catalysis, or the T state, which reduces it. R and T states behave much more like dimmer switches, as I noted, than on-off switches, with the R state corresponding to the higher activity and the T state corresponding to the lower activity. If we look next at the behavior of one particular allosteric enzyme, you'll see how much such controls allow an enzyme activity to be tailored in response to cellular needs. The example we're going to focus on is a very well-studied enzyme that brings together two substrates to make a building block for DNA. As you probably know, DNA is made from 4 kinds of subunits called nucleotides, and they differ only in the component called a base. The 4 kinds of bases in DNA are adenine, thymine, cytosine, and guanine, usually abbreviated as A, T, C, and G. Nucleotides carrying the C or T are called pyrimidine nucleotides, and nucleotides that carry the A or G are called purine nucleotides. Every step in the ladder-like structure of DNA contains one purine and one pyrimidine, with A always paired with T and G always paired with C. When cells copy DNA, they need supplies of all 4 nucleotides in the correct amounts, so that enzymes that make these nucleotides must be very carefully regulated. Purines and pyrimidines always occur in pairs in DNA, so you don't want too much of one kind any more than you would want, let's say, more left shoes than right ones. Our enzyme of interest is known as ATCase, and what it does is catalyze an early step in the pathway that ultimately makes a nucleotide with cytosine called CTP. If there's already plenty of CTP, cells need to ensure that the ATCase containing pathway doesn't keep making more CTP and other pyrimidines. They also need to balance the production of pyrimidines like cytosine with purines such as adenine. So if there's high levels of purines, we want to increase ATCase activity to make enough pyrimidines to balance them out. So control of enzyme activity depends on two things: being able to sense the levels of the different nucleotides, and then being able to increase or decrease enzyme activity in response to those levels. Let's see how the cell handles regulation of this enzyme. Each ATCase enzyme is made up of 12 polypeptide subunits. 6 of the polypeptides in ATCase catalyze the reaction at their active sites. The other 6 polypeptides of the enzyme regulate the activity of the enzyme. Now, all of the subunits work together like a thermostat on a furnace regulating its own output. The regulatory subunits have a binding site called the allosteric or effector binding site. But instead of substrate, these sites bind CTP, which represents team pyrimidines in our scheme. Or else it binds ATP, representing team purines. Now, let's see how the binding of these molecules to the allosteric site affects the enzyme's activity. When the cellular levels of the pyrimidine CTP are high, it latches on to the allosteric sites on the regulatory subunits of ATCase. Binding of CTP to those sites shifts the enzyme into the T state, reducing its affinity for substrate. Reduced binding of substrate in turn dials down the other products needed for synthesis of CTP. In this situation, CTP is said to be a negative allosteric effector, as it is decreasing the activity of the enzyme. On the other hand, if levels of ATP are high relative to CTP, then ATP binds to the enzyme. Now, how does the enzyme know which nucleotide is more abundant to maintain the purine-pyrimidine balance? It doesn't. This is a case of musical chairs at a molecular basis. If there are more ATP molecules than CTP, then ATP molecules are more likely to occupy those allosteric sites. ATP binding does the opposite of CTP binding, shifting the enzyme into the R state, which has higher affinity for substrates. Binding of more substrate increases the synthesis of the product needed to make CTP and other pyrimidine nucleotides. Here, ATP is acting as a positive allosteric effector because it's increasing the activity of the enzyme. The beauty of this dual regulation is that CTP indicates levels of pyrimidines and ATP that of purines. The one present in the greater concentration controls ATCase and the entire pathway. When pyrimidines are higher, ATCase and the pathway get turned off. When purines are higher, then synthesis of pyrimidines gets turned on. This is classic allosteric regulation. Enzyme activity is turned up or down depending on the concentration of molecules in the cell. One more lesson to learn from ATCase, and it's an important one for control of metabolic pathways. ATCase does not directly make CTP. It makes a molecule that is later converted into CTP by the pathway. CTP binding to ATCase and inhibiting its activity stops the synthesis of the very first molecule in the pathway and thus stops the entire pathway. It's much like an assembly line where stopping the first worker stops all the production. This is a classic case of feedback inhibition. Allosteric regulation is reversible, and it depends on the concentration of allosteric regulatory molecules. High concentrations are necessary for occupying the allosteric sites. Occupation of the allosteric site is what modulates the enzyme's synthesis of product. Allosteric controls are great if what you want to do is control the volume of the reaction up or down. What if you prefer an actual on-off switch? Well, one kind of on-off switch acts like a sealed for your protection package. Until you break the seal, you can't get at the goodies. And the seal in this on-off switch is a peptide bond that blocks access to the active site. Enzymes that start with the active site being covered up like this are known as Zymogens or proenzymes. The inactive form of an enzyme like lipase or trypsin will have a name like prolipase or trypsinogen to tell you it's not the active form. It's only after one or more peptide bonds in the covered enzyme are broken that substrates get access to the active site. So really this is an off-on switch, since the zymogen is always off until activated by bond cleavage. The term zymogen comes from zyme, meaning enzyme, and gen, meaning give rise to. Many digestive proteases are made as zymogens. Zymogens are one way to keep enzymes inactive until needed. Now, I mentioned chymotrypsin earlier, which acts as a protease in intestines, but will attack the pancreas that makes it if it's activated too soon. Chymotrypsin is made in a form called chymotrypsinogen, which is inactive, and is only referred to as chymotrypsin after the active site is exposed by peptide bond cleavage. An important set of enzymes known as zymogens are those involved in the formation of blood clots. Blood clotting poses formidable challenges for the body. On the one hand, the blood supply is limited, so cuts must be quickly fixed or an individual will bleed to death. However, if the clotting system is too active, clots may form within the blood supply itself and stop the flow of blood. This can lead to the painful condition thrombophlebitis, or worse, heart attack or stroke. To balance these challenges, the body makes all of the necessary proteins for clot formation and releases them in the blood, so they're ready to go into action instantly when needed. But so that clots don't form routinely, all these enzymes and proteins travel in the blood as zymogens. They can't form clots without activation. The system is beautifully adapted to its task. Upon the detection of a wound, zymogens are activated in a precise and sequential fashion, not unlike a set of dominoes where the falling of one causes the falling of the next ones. Each enzyme in the series is a protease, which is activated by peptide bond cleavage and then proceeds to activate the next enzyme in the same way. The ultimate goal of the clotting pathway is to convert an inactive protein called fibrinogen to an active form called fibrin. Fibrinogen is inactive because it cannot form polymers that would form a clot, where the active protein fibrin is able to form a polymerized clot that stops the bleeding. So if speed is of the essence, why does the body have multiple steps activating the clot-forming protein? Well, at each step in the process, there's an amplification of the response, making it both swifter and more overwhelmingly effective. And enzyme action, as you know, is blindingly fast. So even with several steps, the entire sequence can happen very, very quickly. For example, consider a system where one molecule of the first enzyme in the series activates 100 molecules of enzyme number 2. Then those 100 molecules of enzyme 2 activate 100 times 100, or 10,000 molecules of enzyme number 3. In short order, a single active molecule of the first enzyme has led to the activation of 10,000 molecules of the third enzyme in the sequence. Blood clotting works in this way, through two different pathways that converge, ultimately making fibrin. And fibrin makes fibers so tightly intertwined that water and blood cannot pass through them. Remarkable. The network of fibrin molecules reinforces the initial plug created by platelets to stop the loss of blood. So thanks to the enormous amplification that occurs at each step, large amounts of fibrin are formed quickly and efficiently in response to a very tiny signal. That, in fact, is how bleeding from a cut on your finger gets stopped in a minute or so. Now, some people lack a specific protein factor in the clotting system called factor VIII, and they suffer from hemophilia, a condition in which blood does not clot properly. Before treatments existed for hemophilia, people with the condition often died by bleeding to death from very minor injuries. Hemophilia was called the royal disease because it affected the royal families of Europe, being passed on through the marriages between them. Prince Leopold, a son of Queen Victoria, for example, died at the age of 30 from a hemorrhage. Fortunately, today, the missing factor VIII can be supplied to hemophiliacs to allow them to live a normal and healthy life. We've seen that the breakage of peptide bonds is important in our bodies to quickly activate proenzymes or zymogens. That would not be safe if always in their active form. But since binding peptide bonds to activate the enzymes is irreversible, you might be wondering how such enzymes can be turned off. That gets us to another control mechanism for enzymes, using other proteins to cover the active site. Proteins called protease inhibitors, for example, inhibit the activity of protease enzymes by binding to their active sites tightly and preventing further catalysis. Though this might seem like the cover that was removed by the protease cleavage, inhibitors are separate peptides and physically project into or disrupt the action of the active site. The protease inhibitor alpha-1 antitrypsin, for example, helps to regulate several proteases, including one in our lungs called elastase. In non-smokers, elastase is controlled properly by this inhibitor. In smokers, however, the alpha-1 antitrypsin gets oxidized at the point where it normally interacts with the elastase, and when this happens, it can no longer control elastase's activity. As a result, the elastase begins attacking lung tissue. And when this happens, emphysema can result from lungs damaged by elastase, similar to what happens when chymotrypsin damages the pancreas. Another common mechanism for enzyme regulation also acts like an on-off switch, but in this case, the switch is reversible, without needing an inhibitor protein to cover the active site. Now, how it works depends on the covalent addition or removal of what is usually a phosphate to the enzyme. These enzymes exist in two states, without a phosphate or with a phosphate. Switching between the two states determines whether it is active or inactive, though which is which depends on the specific enzyme. That is, some enzymes are in the on when the phosphate is linked to them, like glycogen phosphorylase. Other enzymes are on when the phosphate is removed, like glycogen synthase. Enzymes controlled by this mechanism get phosphate groups attached to amino acid side chains that are usually the polar but uncharged side chains of serine, threonine, or tyrosine. Special phosphate-adding enzymes called kinases covalently link a phosphate group to the appropriate side chain in a process called phosphorylation. Phosphate groups are negatively charged, so adding them to the uncharged side chain changes them from neutral to negative. And as you might imagine, that affects the forces that stabilize protein structure. For example, if a negatively charged aspartic acid side chain is next to a serine, and that serine gets changed to a negatively charged phosphate, the aspartic acid side chain will be repelled by the phosphate, where it was not repelled by the serine. Now, this could cause a small movement and make a subtle change to the enzyme's conformation. Since function is related to structure, the new structure might be active where the previous one was inactive, or vice versa. So think of light switches. Even just a small movement can cause them to go on or off. However, unlike allosteric regulation, where the binding of the regulatory molecule is non-covalent and readily reversible on its own, phosphorylation can only be reversed by removing the phosphate group using another enzyme called a phosphatase. So the reversible switch needs two enzymes. One enzyme is needed for phosphorylation and another to remove the phosphate. Putting phosphates onto things is why we have kinases, and enzymes taking phosphates off are protein phosphatases. If the enzyme is turned on when it's phosphorylated It gets turned off when the phosphate is removed. On the other hand, if it was active in the absence of the phosphate group, addition of the phosphate will turn it off. So enzymes can toggle between their active and inactive states using phosphorylation and dephosphorylation. A typical mammalian cell contains thousands of kinds of phosphorylated enzymes. And switching those enzymes back and forth are several hundred kinases and phosphatases, which are often responding to neural and hormonal signals. Many familiar drugs in use today work by changing the activity of a specific enzyme. Even some ancient herbal remedies like willow bark, long used to treat pain, work because they contain compounds that inhibit the activity of an enzyme. Willow bark, we now know, contains a compound called salicin, which in the body is converted to a molecule similar to aspirin. Aspirin blocks an enzyme called cyclooxygenase 2 from making molecules that cause pain and inflammation. The majority of enzyme regulators used as drugs work by reducing the activity of the target enzyme. Enzyme inhibition by drugs involves molecules binding to enzymes, reversibly or irreversibly. Now, the irreversible ones are sometimes called suicide inhibitors. These inhibitors usually resemble the enzyme's normal substrate, but once bound, the inhibitor forms a covalent bond to the active site, blocking it permanently, hence the suicide. A common suicide inhibitor is penicillin. It acts by binding to a bacterial enzyme responsible for making bacterial cell walls. Unable to complete the synthesis of a functional cell wall, the bacteria die. Now, unlike penicillin, though, most enzyme inhibitors used in medicine are reversible because many might act on our own enzymes and we don't want to permanently disable them. Reversible inhibition comes in several forms on a spectrum from competitive to partially competitive to non-competitive and uncompetitive. But the most common of these for medical purposes is called competitive inhibition, and we're going to focus on that. In competitive inhibition, the inhibiting molecule resembles the normal substrate of the enzyme and competes with it for binding at the active site. When the inhibitor binds to the enzyme, the normal substrate cannot, and the enzyme is inactive. This is similar to what we saw for suicide inhibitors with one crucial difference. The competitive inhibitor does not make covalent links to the enzyme. With no connection between the enzyme and the inhibitor, their interaction is temporary and readily reversible. Now, the extent of inhibition depends on the relative concentrations of the inhibitor and the substrate. Since the inhibitor molecules and the substrate molecules are aiming for the same active site, much like ATP and CTP were competing for the same allosteric binding site of ATCase. If there are more substrate molecules than inhibitor molecules, the chance that the active site will bind substrate will be higher. Conversely, if large numbers of inhibitor molecules and few substrate molecules, the odds of the inhibitor molecules occupying the active site are higher. Thus, in competitive inhibition, addition of more substrate results in decreasing the extent of inhibition. If the substrate concentration is high enough, the inhibition can be completely overcome. The approach to reversible enzyme inhibition that is typically attempted in drug design is competitive inhibition, as I said. Because competitive inhibitors bind the active site, such drugs can be designed to resemble the substrate of the target enzyme. On the other hand, mimicking the substrate simply involves clever chemistry. By contrast, reversible inhibition that is non-competitive or uncompetitive is not commonly used medicinally. These inhibitors used in these mechanisms do not resemble the substrate and do not bind to the active site, so it's harder to figure out what such an inhibitor would look like. An excellent example of synthetic drugs acting as competitive inhibitors are the statin drugs, used by over 22 million Americans to lower blood cholesterol. Statins lower the activity of an enzyme called HMG-CoA reductase that plays a key role in cholesterol synthesis in our liver. The normal substrate of HMG-CoA reductase is called HMG-CoA. The structure of HMG-CoA is mimicked in the drug lovastatin, more commonly known as Lipitor. Because of the similarity between the normal substrate, which is HMG-CoA, and the drug, lovastatin, lovastatin acts as a competitive inhibitor, occupying the active site and preventing the enzyme from acting on its substrate. As a result, less cholesterol is made in the body. All these and many other examples highlight why understanding enzyme regulation is invaluable in developing drugs. So let's wrap this up. Hundreds of drugs have been designed based on our knowledge of how enzymes are regulated. These include antiviral drugs like Ara-A that fool the DNA polymerase of herpes viruses, and protease inhibitors that stop HIV. Because of the pivotal roles enzymes play in metabolic pathways, the more we know about what controls enzymes, the more effective our therapies will be. Your LDL levels are high. The doctor told me with a sigh. She will treat the condition with competitive inhibition, because if we don't, I may die.